In the summer of 1991, a team of archaeologists excavating a burial site in the Kenyan Rift Valley uncovered the skeletal remains of a Homo sapiens individual who had lived approximately 40,000 years ago. The bones were remarkable not for what they showed, but for what they didn't. No signs of the chronic inflammatory conditions that now affect roughly half the adult population of the industrialized world. No evidence of the autoimmune lesions that leave characteristic marks on joint tissue and bone. No skeletal indicators of the metabolic dysfunction that produces the specific bone density patterns associated with insulin resistance and chronic systemic inflammation. This was not an isolated finding. Across the entire corpus of well-preserved Paleolithic skeletal remains, thousands of specimens from sites across Europe, Africa, Asia, and the Americas, researchers studying the evidence of immune-mediated disease have found the same consistent absence. The chronic inflammatory diseases that now constitute the single largest category of medical expenditure in the developed world are essentially invisible in the prehistoric record. Rheumatoid arthritis, multiple sclerosis, inflammatory bowel disease, lupus, type 1 diabetes, psoriasis, Crohn's disease. The conditions that affect an estimated 1 in 12 people in industrialized nations, conditions in which the immune system attacks the body's own tissues, were not the dominant health reality of our species for the first 300,000 years of its existence. They are a recent development, and the timeline of their emergence corresponds precisely with a specific set of behavioral changes that occurred in the last century. Not genetics, behavior. Here is what the immunology textbooks almost never say directly. The human immune system is not a fixed biological asset. It is a trained system, a learning system. A system that requires continuous exposure to a specific and diverse range of microbial and environmental inputs to develop, calibrate, and maintain the discrimination capacity that separates effective immune defense from the catastrophic misfiring we call autoimmune disease. For 300,000 years, that training happened automatically. Every hand that touched soil, every root pulled from the ground and eaten without sterilization. Every breath of outdoor air carrying microbial diversity that no indoor environment replicates. Every fever allowed to run its course. Every childhood infection resolved without pharmaceutical suppression. The training was continuous, involuntary, and relentless. And then, in the space of approximately 100 years, we removed it. We did not intend to disarm the most sophisticated biological defense system evolution ever produced. We were trying to stay clean. We were trying to manage fever. We were trying to prevent infection. The consequences of that effort are now the dominant chronic disease pattern of the developed world. And the prehistoric skeleton lying in a Kenyan burial site 40,000 years after death is the evidence that something has gone profoundly wrong. Before we continue, I want you to think about something. Think about the last time you were genuinely sick, a real infection, fever, immune response, the full biological event. Not a sniffle managed with antihistamines, a real fight. And then think about what happened after. The recovery. The specific clarity that follows a genuine immune response. The feeling of a system that has done its job and returned to baseline. Now think about how often, in your modern life, you have interrupted that process. How many fevers you have suppressed with ibuprofen before they could complete their function. How many surfaces you have sterilized that did not need sterilizing. How many courses of antibiotics you took for conditions that your immune system could have resolved without pharmaceutical intervention. I am not making a moral argument. I am asking you to hold a question that the chronic disease data is asking whether you are prepared to answer. What if every intervention you made to avoid the discomfort of a normal immune response was simultaneously preventing the training that makes the next immune response more precise, more efficient, and less likely to misfire on your own tissue? Here is the theory that immunologists have been quietly building the case for since the late 1980s, and that has become, in the past decade, one of the most well-supported frameworks in all of modern medicine, despite remaining almost entirely absent from public health messaging. The theory was first articulated in 1989 by epidemiologist David Strachan in a paper published in the British Medical Journal, and it was initially called the hygiene hypothesis. Strachan noticed that children who grew up in larger families with more siblings, and therefore more early childhood infections, had significantly lower rates of allergic disease than children who grew up in smaller, cleaner, more isolated households. His hypothesis was simple. Early exposure to infection trains the immune system. The absence of that exposure leaves the system undertrained, overreactive, and prone to misfiring on harmless antigens. The original hypothesis has been substantially revised and expanded in the decades since. The current framework, called the Old Friends Hypothesis, developed primarily by immunologist Graham Rook at University College London, is more precise and more disturbing. It is not infections per se that train the immune system. It is the ancient microbial organisms that co-evolved with the human immune system over hundreds of thousands of years, organisms found in soil, in untreated water, in fermented foods, in the gut microbiomes of animals our ancestors lived alongside, and in the human microbiome itself, whose presence regulates the immune system's inflammatory response. Remove those organisms, and the immune system loses its primary regulatory input. It does not go quiet. It goes hyperactive. In 2003, immunologist Joel Weinstock at Tufts University published a paper in the journal Gut that produced one of the most counterintuitive findings in the history of modern medicine. Weinstock and his team had been studying the epidemiology of inflammatory bowel disease, specifically the striking pattern that IBD rates were dramatically lower in regions with high prevalence of intestinal parasites, and dramatically higher in the same regions after parasites were eliminated through improved sanitation. The correlation was so consistent that Weinstock formed a hypothesis. Perhaps the parasites were not incidental to gut immune regulation. Perhaps they were part of it. He ran a small clinical trial in which patients with treatment-resistant Crohn's disease were given controlled doses of Trichuris suizova, pig whipworm eggs, harmless to humans, to swallow. The results were remarkable and deeply uncomfortable for anyone committed to the simple narrative that parasites are always enemies. 72% of the treated patients showed significant clinical improvement. Several went into remission. The introduction of an ancient microbial organism that the human gut immune system had coexisted with for hundreds of thousands of years appeared to restore a regulatory function that its absence had disrupted. Weinstock was not advocating for parasitic infection as a health strategy. He was demonstrating something that the data kept insisting on. The human immune system evolved in the presence of specific microbial inputs, and their removal has consequences that pharmaceuticals are struggling to replace. Wait, if the introduction of a parasite can reduce the symptoms of an autoimmune disease, what does that tell us about what the ancestral immune system was actually calibrated to do? But the parasite finding is only the most dramatic piece of this story, because the research on ancestral immune calibration goes much further than intestinal parasites, and the mechanisms involved are more fundamental, more pervasive, and more directly connected to behaviors that the modern world has systematically eliminated. The full picture requires going into the soil itself, into the specific organisms that live there, into what happens when a human hand contacts that soil daily for decades, and what has happened to the immune system that has not contacted it in a generation. Let's follow the evidence like detectives. The soil microbiome, the community of bacteria, fungi, archaea, and other microorganisms living in the top layer of Earth, is one of the most diverse biological environments on the planet. A single gram of healthy topsoil contains approximately 1 billion bacterial cells, representing 10,000 or more distinct species. For most of human evolutionary history, that community was in constant contact with the human body, through food grown in unsterilized soil and eaten without industrial washing, through hands that touched ground daily during foraging, tool making, shelter building, and every other physical activity of prehistoric life, through water sourced from the ground that carried soil microorganisms into the digestive system, through the simple act of sleeping on the earth, which transfers microbial diversity to skin continuously. One organism from this community has been studied with particular attention by immunologists over the past 2 decades, Mycobacterium vaccae, a harmless soil bacterium that is ingested and inhaled routinely by anyone working with or near healthy soil. Research beginning in the early 2000s and accelerating substantially in the 2010s has demonstrated that M. vaccae has direct and measurable effects on the human immune system and nervous system that go far beyond simple immune training. A 2004 paper by oncologist Mary O'Brien found that lung cancer patients who received injections of killed M. vaccae showed significant improvements in quality of life, emotional well-being, and cognitive function compared to control groups, effects that were not attributable to any anti-cancer mechanism. A 2016 study by neuroscientist Christopher Lowry at the University of Colorado Boulder found that M. vaccae activates specific neurons in the raphe nuclei, the brain region responsible for serotonin production, producing measurable anxiolytic and antidepressant effects in animal models. The soil bacterium that prehistoric humans encountered daily through physical contact with the earth was not just training their immune systems. It was modulating their neurological stress response. It was, in a very specific biochemical sense, making them calmer and more emotionally resilient. The people digging in the soil every day were not just gathering food. They were, without knowing it, administering a daily dose of a neurologically active, immune regulatory organism that their biology had spent hundreds of thousands of years learning to use. The old friends hypothesis has accumulated substantial empirical support across multiple independent research programs. A landmark 2012 study published in Science by researchers at the Brigham and Women's Hospital found that germ-free mice, animals raised in completely sterile conditions with no microbial exposure, showed dramatically elevated levels of invariant natural killer T cells and significantly increased inflammatory responses in the gut and lungs compared to conventionally raised mice. When the germ-free mice were colonized with normal gut bacteria in the neonatal period, their immune profiles normalized. When colonization was delayed until adulthood, the dysregulation persisted. The critical window for immune calibration was early life exposure. The germ-free mice were not immunodeficient. They were immune dysregulated. Their systems produced excessive, misdirected inflammatory responses because the regulatory inputs provided by microbial colonization were absent. A 2016 paper in the New England Journal of Medicine analyzing health outcomes in the Amish and Hutterite farming communities of the American Midwest provided a striking human analog. Despite similar genetic backgrounds and similar lifestyles in many respects, Amish children, who grow up on traditional single-family farms with direct daily contact with farm animals, soil, and raw agricultural environments, showed asthma rates of approximately 5%. Hutterite children, whose communities use large-scale industrial farming with significantly less direct animal and soil contact, showed asthma rates of approximately 21%. The difference was not genetic. The researchers performed house dust analysis and found that Amish homes contained dramatically higher concentrations of diverse microbial products, endotoxins, bacterial cell wall components than Hutterite homes. The children whose immune systems were being continuously exposed to diverse microbial input from their physical environment showed a fraction of the inflammatory misfiring of children living in cleaner, more microbiologically impoverished environments. Think about what the ancestral immune calibration environment actually looked like at the level of daily life. A Paleolithic child growing up in a band of 20 people in grassland Africa was, from the moment of birth, colonized by the microbial community of their mother, their family, and their physical environment simultaneously. They were nursed with milk that contained not just nutrition, but a specific prebiotic complex, human milk oligosaccharides, that fed specific gut bacterial populations associated with immune regulation. They crawled on soil, they put objects found on the ground in their mouths. They ate food grown in soil without washing it under treated water. They were bitten by insects that transferred environmental microorganisms into the bloodstream. They lived in close physical contact with animals whose microbiomes contributed to the diversity of their own. And when they got sick, a fever, a respiratory infection, a skin infection, the fever was allowed to run. Not because their community was indifferent to their suffering, but because there was no mechanism to suppress it, and because the fever itself is an immune response, not a symptom. The elevated temperature of a febrile response inhibits bacterial replication, enhances immune cell function, and accelerates the resolution of infection. The child who survived that fever had an immune system that had experienced the complete cycle of pathogen detection, response, resolution, and memory formation. The immune memory formed by that experience, the specific antibodies, the trained T-cell populations, the innate immune adaptations, would remain for decades. In some cases, for a lifetime. The child who experienced that same infection suppressed with antipyretics and antibiotics at its first sign experienced a truncated version of the same encounter. The pathogen was cleared, but the immune education was incomplete. The memory was weaker. The calibration was shallower. I want you to feel what the ancestral immune environment actually felt like from the inside. You are 6 years old. Your feet are bare on earth that has never seen a synthetic chemical. Your hands are dirty in a way that no parent in the modern world would permit. The food you ate this morning was pulled from the ground and rubbed clean on your clothing. You have a runny nose. You have had a runny nose, off and on, since you were old enough to crawl. Your body runs warm sometimes, not sickeningly hot, but warm, the way a well-used machine runs warm. The adults around you do not panic at this warmth. They watch you. They give you water. They keep you close to the fire. And then the fever passes, and you feel the specific aliveness that follows, the clarity of a system that has just won a fight and knows exactly what it defeated. By the time you are an adult, your immune system has encountered dozens of pathogens, hundreds of environmental microorganisms, thousands of microbial inputs from soil and water and air and physical contact with the living world around you. It has trained against all of them, not perfectly, not without cost, but thoroughly. The system running in your body at 30 years old is a calibrated instrument, not calibrated by genetics, not calibrated by medicine, but calibrated by continuous, unmediated contact with the microbial world. And when a new threat arrives, the system responds with the precision of something that has been in active training for 3 decades. Not because you were born different, because you lived differently. But the soil bacteria and the early life infection story are only the beginning. Revelation 1. A 2019 study published in Science by researchers at the Broad Institute analyzed the gut microbiomes of 61 individuals from the Yanomami people of the Venezuelan Amazon, one of the most isolated indigenous populations on Earth, with essentially no contact with the modern medical system, and found microbial diversity levels approximately 50% higher than those of age-matched American adults. More significantly, the Yanomami microbiomes contain functional gene categories associated with antibiotic resistance that have never been documented in any modern antibiotic, suggesting that the microbial ecosystem of the human gut had been co-evolving with environmental antibiotics for far longer than the pharmaceutical era. The modern gut is not a depleted version of the ancestral gut. It is a fundamentally restructured one. Revelation 2, a 2018 paper in Cell Host and Microbe, found that the administration of broad-spectrum antibiotics in early childhood, specifically in the first 2 years of life, produced measurable and persistent alterations in gut microbial composition that were still detectable 5 years after the antibiotic course ended, and that correlated with elevated rates of allergic disease, asthma, and inflammatory bowel conditions in the children who received them compared to children who did not. The antibiotic did not just treat the infection, it restructured the training environment of the immune system for years afterward. Revelation 3, and this is the one that reframes everything, a 2021 meta-analysis published in The Lancet analyzed the global epidemiology of autoimmune disease rates from 1990 to 2019 and found that every single autoimmune condition studied was increasing in incidence in every region of the world undergoing sanitation transition, the process of moving from traditional to modern hygiene standards. The autoimmune epidemic is not a genetic phenomenon. It is not a detection artifact produced by improved diagnostics. It is a real, accelerating, globally consistent increase in immune misfiring that tracks with a single behavioral variable: the removal of ancient microbial inputs from the human biological environment. Here is the question that the public health establishment has been structurally unable to ask directly, because the answer implicates interventions that have saved tens of millions of lives. If the evidence clearly shows that the removal of early life microbial exposure, the suppression of fever responses, the disruption of gut microbiome diversity by antibiotic use, and the elimination of soil and environmental microbial contact are driving the autoimmune epidemic, if these specific modern behaviors are producing measurable, reproducible, mechanistically explained immune dysregulation, then what is the correct public health response? You cannot uninvent antibiotics. You should not withhold them from a child with bacterial meningitis. You should not allow children to die of preventable infections to preserve their immune training environment. But you can ask whether the same interventions that are essential in life-threatening infections are being applied at a scale, in livestock, in agriculture, in routine childhood medicine, in food processing, in environmental sterilization, that goes far beyond necessity and produces immune consequences that are costing more lives in the long run than they are saving in the short term. That is not a comfortable question. It is the question that the autoimmune data is insisting on. And it is the question that the ancestral immune system, calibrated over 300,000 years, running cleanly in a world that no longer exists, is asking every time a modern person develops a condition in which their immune system attacks their own joints, their own gut, their own nervous system, for reasons that medicine can describe but cannot yet cure. But here is where our modern understanding of immune health completely fails us. We think about immunity in terms of defense, strength, the ability to fight pathogens and win. We measure immune health by absence of infection. But the ancestral immune system was not just a weapon, it was a diplomat. A system that had learned, through 300,000 years of continuous negotiation with the microbial world, exactly which organisms were threats, which were neutral, which were beneficial, and which were ancient partners whose presence it depended on for its own regulatory stability. The modern immune system, stripped of that negotiating history, cannot make those distinctions as reliably. It sees threats everywhere. It fires at harmless pollen. It attacks joint tissue. It inflames the gut wall. It is not weak. It is confused. And the confusion is the direct result of removing the education that gave it context. Here is the assumption most people bring to this topic, that the answer to immune dysregulation is to be exposed to more pathogens, to get sick more, to let infections run their course as a form of immune training. The science does not support this framing, and it is important to say so clearly. The old friend's hypothesis is not an argument for infectious disease as a health intervention. The organisms that calibrate the immune system are not primarily pathogens. They are the ancient microbial partners, soil bacteria, gut commensals, environmental fungi, the organisms that live on skin and in water, and in the ecosystems that our ancestors inhabited continuously. Many of them are harmless in themselves. Their role is regulatory, not adversarial. The immune system was not calibrated by surviving dangerous infections. It was calibrated by continuous, low-level, diverse exposure to a microbial world that it had co-evolved with for hundreds of thousands of years. The practical implication is not let your children get sick. It is something both simpler and more systemic. Put your hands in soil. Eat food with some relationship to the ground it came from. Spend time in environments that are not sterilized. Do not suppress every fever that your immune system generates in the course of doing its job. And when antibiotics are genuinely necessary, use them, but understand that every unnecessary course is a disruption to a microbial ecosystem that your immune system uses as its primary training ground. This is what 300,000 years of immune calibration data has been building toward. The Kenyan skeleton with no autoimmune lesions. The Yanomami gut microbiome with 50% greater diversity than a modern American adult. The Amish children with 5% asthma rates living on traditional farms. The germ-free mice whose immune systems cannot be corrected once the critical calibration window closes. The Trichuris Suze trial in which a harmless ancient parasite reduced Crohn's disease symptoms in 72% of treatment-resistant patients. The global epidemiology showing every autoimmune condition rising in every region undergoing sanitation transition. Taken together, these data points describe not a collection of separate phenomena, but a single, coherent biological reality. The human immune system is a learning system that requires specific inputs to function correctly. Those inputs are ancient, diverse, microbial, and environmental. They were present in the daily lives of every human being who lived before approximately 100 years ago. They are systematically absent from the daily lives of most people living in industrialized nations today. The immune systems running in modern bodies are not malfunctioning. They are running without their training data. And the chronic inflammatory diseases that now affect hundreds of millions of people globally are the output of immune systems that were built to negotiate with a microbial world they have never encountered. The research on behavioral interventions for immune recalibration has converged on findings that are more specific and more actionable than most popular health communication acknowledges. A 2019 study in Science of the Total Environment found that children who spent at least 90 minutes per day in natural outdoor environments showed significantly greater gut microbiome diversity and lower inflammatory markers than children whose outdoor time was primarily in paved or manicured urban spaces, not because forests are magic, but because the microbial diversity of natural soil, vegetation, and air differs profoundly from the microbial poverty of sanitized urban environments. A 2020 paper in Gut found that individuals who consumed a diet containing 30 or more distinct plant species per week, raw, fermented, minimally processed, showed significantly lower inflammatory cytokine profiles and greater immune regulatory function than individuals consuming fewer than 10 plant species per week, regardless of total caloric intake. And every time you use an antibacterial hand sanitizer on hands that are not at epidemiologically meaningful contamination risk in your own home, after touching objects in your own environment, in contexts where no pathogen exposure has occurred, you are suppressing the skin microbiome that functions as one of the immune system's peripheral training environments. Not dramatically, not catastrophically, but incrementally. Every unnecessary application is a small withdrawal from an account that your ancestral immune system spent 300,000 years building, and that your biology depends on to maintain the calibration that distinguishes your own tissue from an enemy. The prehistoric human who lived in continuous contact with soil, with animals, with outdoor air, with the full spectrum of the microbial world, who allowed their fevers to complete their function, who ate food with living microbial communities still attached, who maintained a gut microbiome of extraordinary diversity, that human was not healthier because they were tougher. They were healthier because the environment they lived in gave their immune system what it needed to do its job correctly. The immune system you carry is the same system. The hardware has not degraded. The 300,000 years of evolutionary refinement that produced it are still present in every cell. What has changed is the software input, the continuous microbial exposure, the diverse gut ecosystem, the unmediated contact with the living world that the system requires to run the programs it was built to run. You cannot recreate the Paleolithic microbiome in a modern city, but you can move toward it. You can put your hands in soil. You can eat fermented food. You can spend time in natural environments. You can resist the instinct to sterilize every surface. You can allow a mild fever to do the work it evolved to do before you suppress it. None of these things are extreme. All of them are ancient. And the immune system running in your body right now is waiting, as it has always been waiting, for the inputs that make it whole. 300,000 years ago, a human child touched soil, breathed outdoor air, ate food that had not been sterilized, ran a fever that was not suppressed, and lived inside a microbial world that their immune system used as a continuous dynamic, endlessly specific training environment. Their immune system learned to distinguish friends from enemies, harmless from dangerous, self from other, with a precision that modern immunology is only now beginning to map. We inherited that system. We live inside a world that has removed almost every input that system evolved to receive. And we are experiencing, in the bodies of hundreds of millions of people, the outcome of that removal, not as a genetic tragedy, not as bad luck, but as the predictable, mechanistically explicable result of a learning system running without its data. So here is the question that the Kenyan skeleton, and the Yanomami microbiome, and the Amish children, and the germ-free mice, and the Trichuris suis trial are all asking simultaneously. The most sophisticated immune system in the history of the animal kingdom is running in your body right now. It was built for a world of soil and microbes, and fever and ancient partners. You gave it hand sanitizer and a sterile apartment and suppressed every fever before it could finish its sentence. What do you think it learned?